Okay, I am John Beckmann, Professor John Beckmann, Dr. John Beckmann, the father of Sid, the transgenic mother of sin, your hostess with the mostest. I want to try to do something unique today. I don't know if anybody's ever done this. I think it's an intriguing concept. So I'm going to try to do like an open air research project. I've never done this before. So what I'm going to do is I have this idea. I want to complete this idea. It's probably going to take me 6 months to a year to complete. And I'm gonna like publicly do like a development vlog on this research project that I want to work on. So normally I would never do this because I've just had really, I feel like really bad. I'm sure most people feel this way. I just feel like the science field is really plagued with people who just like steal ideas. And so I thought it would be really interesting to take one of my projects and to try to do it completely publicly in the open as I go through it. So again, this is literally like kind of like the first week that I started working on this. And I think it'll be funny because, well, one, okay, I would never do this with like one of my high impact studies. And I have some high impact studies in the pipeline, which I would be super concerned about just publicly like releasing those. But I'm at a point now where that research is solidified. It's going to be coming out soon. I'm less worried about that. And I kind of want to now just do some fun stuff. Like I really want to do some fun projects. And this project that I want to work on is not something that is going to be super high impact, and I don't think it's anything that somebody would steal. It would take them a lot of work to steal this, and I think it would be funny if they did, because if they did, I'm in a position now where I can call them out on it, and I can say, you fucking stole this project. So I don't think anybody's going to do that. So, which is one reason why I would not advise anybody else to do this. But I'm in a position with this project that I don't think not many other people can do this or would be interested in it even, or would be even interested in stealing it. So I think this is going to be fun to publicly do this. So more context on this project. So what I want to work on is I want to start combining my computer science background with my CI background, my CI research. So if you didn't know, I have been, since I started my assistant professorship, I have been pursuing a second undergraduate degree in computer science. I have taken about 8 classes now in the computer science field. So I feel I'm no longer an amateur. Like I definitely, like, I'm not an expert for sure, but I know enough to be dangerous, and I know enough to like do stuff. So I want to start like working on projects, which again, like this is not going to be like super high impact. This is not going to be like me solving P versus NP, but this is an opportunity for me to combine my computer science knowledge that I've accumulated thus far in a research project that relates to my faculty position as a professor who researches cytoplasmic incompatibility. So I want to combine these. And the best, so one of the most practical, I think, one of the most useful classes that I've taken so far was Evolutionary Computing. This was just an awesome class, an amazing class. It pushed my Python knowledge to the limits, coding algorithms, my own algorithms, solving problems. It was a heavy project-oriented class. So we actually, at the end of the class, we were literally like, we had constructed a Pac-Man game, and we had constructed Pac-Man controllers and Ghost controllers, and we started evolving the behavior of the Pac-Man controllers in competitive evolutionary algorithms, involving Pac-Man controllers that would play the Pac-Man under some rubric of rules and evolving those rules, and then in tandem co-evolving ghost controllers that would control the ghosts to kill the Pac-Man under a certain rubric of rules and then evolving those rules. So this was like an amazing class. I learned a lot about evolution by literally like coding it. Um, and so shout out to Dr. T who teaches this class, amazing class. If you're in computer science, you should take it. It's super hard, but it was the most intensive class I've ever taken in my entire life. I was coding like 20 I was literally coding like 20 hours a week. My entire weekends were consumed by this class. So you don't have a life if you take this class. But it was an amazing class. It was a lot of fun. And I feel like I learned enough, again, as Dr. T would say, I learned just enough to be dangerous in this field. So I want to start like seeing if I can use it. I have been frustrated with the computer science curriculum in a sense that most of the things that I've taken, I felt like I would never apply this. But this was one class where it's like, I should really start applying this. I should try to start applying it and see what comes up. And I'm not going to be the best, perhaps, well, maybe I would be the best person for this one from the CI aspect, but I'm not going to be the best computer scientist, certainly for sure. I'm not the best coder. But again, as a outlet to improve my coding skills, I like this project. So essentially what we learned in this class was you can design algorithms, evolutionary algorithms or genetic algorithms that essentially like evolve strategies to solve problems. And you can evolve essentially like the strategy or the solution to the point where you can find a good solution. Okay, so this is like I guess a problem-solving tactic. If you don't know how to solve a particular problem, you might code an evolutionary algorithm that you could evolve to try to find a solution to that problem. Okay, so so what I want to do is I want to take what I learned here and apply it to cytoplasmic incompatibility. So I have a really unique system that I study, in a sense that I study these toxin antidote operons. So there's these genes where there's a toxin and there's an antidote. And these genes control and regulate cytoplasmic incompatibility, okay? And this is going to be within the CI playlist. So if you don't know what CI is, like there's really no point in you watching this video. You should go to the CI playlist and start at video 1 if you don't know what CI is. But needless to say, it's a complex field, and the most basal element are these CI operons, okay, that are toxin antidote operons. And these are selfish genetic elements. So my view of them, and okay, you should just a disclaimer, I am not an evolutionary biologist. I'm more of a molecular biologist. I don't have evolutionary biology training. What I know about evolution, as another colleague here would say, I learned on the streets. And I learned from reading Origin of Species by Darwin, reading Dawkins' Selfish Gene, The Ancestor's Tale. Like what I learned about evolution, I learned from like reading kind of like books. So I have not been classically trained per se in evolution. So perhaps you should just not listen to anything I say in this entire presentation or this entire project. But I'll be curious to see at the end of the research project, if I finish it, I'll be curious to see what real evolutionists think about it. And I think I come with a unique angle in a sense that I'm perhaps not biased by the dogmas of that field. I come at this as a student trying to do a research project, trying to learn. That's where I'm coming at this from. So don't even listen to me. Okay, so the CI is this reproductive phenotype, sterilizes insect sperm or rescues the insect sperm. It's controlled by these toxin antidote operons. And the system is super complex. These toxin antidote operons are selfish genetic elements in themselves. So they're going to have their own perspective and their own view, kind of like on evolution. Like they're going to have tweaks of this system that is theirs that they're going to like, okay? But these TA systems themselves, you find them inside plasmids and viruses. Okay, so you find these selfish genetic elements inside of other selfish genetic elements, which are plasmids or viruses. Okay, so they package them in, you can find them in the genetics. If you sequence the genes of certain plasmids in rickettsia, you can find these TA systems on them. And if you, in theory, it's thought that there are viruses in Wolbachia that carry these toxin antidote operons around. So there's a question that I wonder about, which is, when are the TAs useful for the plasmids or the viruses? Like, under what context in evolution or ecology or biology, under what context is it useful for the viruses or the plasmids? Like, does that ever happen? Why do they carry them around? That's kind of a question that I want to start thinking about. And I don't think it's a question that has a hardcore answer yet. Now, it even gets more complex than that. So these viruses and these plasmids live inside other selfish elements, which are the symbiont bacteria. So if this is like a Wolbachia, okay. These Wolbachia have in terms of rickettsia, they have plasmids with the SIFs. The Wolbachia have viruses with the toxin antidote operons. So this is a nested system of selfish elements, okay? There's toxin antidote operons that are inside of plasmids and viruses that live inside of symbiotic bacteria, which themselves live inside of insects and arthropods. So all these, if I can zoom out here, all these elements, they don't necessarily have the same interests, okay? There's one unit of interest here. There's one discrete quanta of interest here. There's one level of interest here, and there's one level of interest here. And it's not clear to me that what helps the TA always helps these other things, and what helps these other things helps the TA. So I want to start getting an understanding of, perhaps this is anthropomorphic, thinking, like putting yourself in their perspective when they're not men and they don't have feelings and they don't have thinking. But I want to start understanding their motivations. Like, what is the goal of these selfish elements? What is the goal of the viruses and the plasmids? What is the goal of the symbiont bacteria, and what are the goals of the insects? And in theory, like according to selfish gene theory, they all want to replicate. Like, that's their goal. They all want to replicate themselves. But I want to start understanding under what context this helps this, or this helps that, or under what context this does not help that, and this does not help that. So again, here's where you should just not listen to me. You should just completely disregard everything I'm saying, because I'm not classically trained. But like the few studies that I've looked at this intensely, so there's like Torelli 1994, which has looked at CI from the perspective of the insect, and for the way that it's phrased, the classical like conclusion from that study would be is that, and here's the way they write it: purifying. Let's zoom in on this so you can see it. Purifying selection does not act on the SIFs. If you want, some call it by that name, some call it by another name, the TA operons. I'm going to use TA operons. You can use it SIFs, whatever the hell you want. Some call it by that name, some call it by a different name. Okay. So in the Torelli 1994 paper, there's this conclusion that is written frequently, and it's sort of my understanding. But my understanding of this, when they say this, purifying selection does not act essentially like to preserve the SIFs. So if you don't know what purifying selection is, like, purifying selection is essentially like if you get a mutation that breaks the gene, this organism would die, and so this would be removed from the population. That's purifying selection. What this conclusion states is that there is no purifying selection. So when these operons break, the organism does not die, and they're perpetuated in the population, which is true. You factually see these broken sieves. You factually see these broken toxin antidote operons, which proves that like purifying selection is not acting to preserve the CI operons. But I don't know like under what perspective that happens. Like they exist, the TA operons exist. So there's some kind of a selection on some level that is selecting for these things to exist. The fact that they are here suggests that this rule is not like all encompassing. And I think the conclusion is sort of like on this level, the level of the insect. The mothers that have these symbionts, that have these viruses, that have these toxin antidote operons, they don't necessarily like need them. Okay. I think that's what the conclusion is. And where you start to see like weird differences is, again, in a previous video I talked about this, like there's a difference in the structure of these operons that you see when you compare Culex versus Drosophila systems, okay? Like in the Drosophila systems, these operons, you see this pattern where they're like breaking, okay? And so it clearly like is evidence, it clearly is evidence of this conclusion from Trelli in 1994, like clearly. And I've done personal work on this that like proves this, like validates this. So I'm not opposed to this conclusion at all. What I don't understand, and again, here's where I'm coming at this from the perspective of a student, what I don't understand is Culex seems to like these things. Like they periodically keep getting duplicated, and you're not seeing completely inactivated duplications. You're seeing what are theoretically like completely active operons that are just like multiplying in Culex. So it seems like there are some contexts under which like perhaps these insects like having these things around. And I'm trying to understand, how does this happen, and how is that different from the death of them when they're dying? Like, what are the evolutionary contexts and trophic levels, I guess? Maybe you say that, maybe that's wrong. Or like, what are the motivations for which these things exist? Like, why are they around if they're not helpful? They must have help under some useful condition. Or maybe they just help themselves, which is why they exist, and they can just sort of like get by. But I can envision scenarios where they're helping a virus and they're helping a plasmid. Certainly, like toxin antidote systems are evolved to select for plasmids. So any plasmid that picks up a TA system is going to be sort of like reproductively more fit than a plasmid that doesn't have it, because it will fucking kill you if you let go of it. So the TA systems are for sure helpful to plasmids, and I would bet that they do the exact same thing to the viruses. If your virus has this thing, it will kill you if you get rid of the virus, okay? And I think that that's how CI evolves. I think that CI evolves from that fundamental mechanism transitioning to the level of the symbiont bacteria. And now it's no longer helping the plasmid, it's helping the symbiont bacteria by manipulating the reproduction of the insects. So at the top level, maybe the insects don't necessarily like need this, but I think that there must be some context under which it's helpful under the bacteria. So I think I can address some of these problems with my computer science background. I can model this. I know for a fact I can model this because we understand the biology enough now to do sort of like complete models of this. And I think I'm the one to do it. I think I know how to do it. Maybe somebody has already done this. I have not even like Googled this research project. If somebody has already done this, I don't even fucking care. Screw you. Like, I don't care if somebody's already done this. I want to try it myself. The point is for me to build the mental infrastructure to understand this problem. And if I just read somebody else's paper and I don't code it for myself, I won't understand it. Plus, I think I can always do things better than other people. So I want to try this for myself. Okay. That is the context of this research project, okay? So that's what I'm going to do. I am going to literally like code Python evolutionary algorithms that act as these players, and I'm going to evolve them in co-evolutionary scenarios, and I'm going to see under which parameters, which values, which parameters sort of like converge on. What are the patterns? Are there any patterns? What does this thing want? What does that thing want? What does the next thing want? What does the next thing want? I think I can answer this with evolutionary computing from what I learned in that class. Shout out, thank you, Dr. T. Although Dr. T just probably does not want to be associated with me. Anyway. Okay. So I've already started working on this, and you can see the first framework from which you start. Okay, the complexity of this is going to grow, and I'm not going to essentially like code a giant program on the first day. You have to start building these elements. So the first element to build are the TA operons and their behavior. Then the second thing to build is the plasmids of the viruses. Then the third thing to build is the symbiont bacteria. And then the fourth thing to build are the arthropods, okay? And then on top of that, there's levels that I'm going to build. I'm going to build essentially like competing populations of insects to sort of think about like, again, I should not say this, I'm not an evolutionary biologist, to think about reproductive isolation mechanisms and things like that. I know that that's kind of like a controversial thing, and I am not like, again, like I'm not the person to debate those controversies, but I want to start thinking about that. It's sort of like speciation and reproductive isolation, and what are the factors involved in that. And then on top of that, geography. So I know from a previous conversation when I did sort of like a deep dive in the Torelli 1994 paper and all the papers that were prior to that, all the modeling papers. Again, I'm not a modeler, don't even fucking listen to me. I'm not an evolutionary biologist, but I did like a deep dive in that research. I felt like I got a good grasp of it. I felt like I understood enough of it. I wrote out like a summary of it. And I remember that I had some conversations with some collaborators, Dr. Charlat, and Dr. Charlat suggested that like one of the things that had not been modeled, and again, this is just from a tidbit from a conversation that I'm thinking of, like one of the things that had not been put into that is geography. So I know that once I build these 1 to 5 layers, I can build a graphical program that will sort of like build blocks, like in the computer science example is the Game of Life, where they have these like cellular automata. I can build these little like pixelated arrays, and I can have sort of like competitive scenarios or draw different sort of geographical boundaries within these pixelated boxes. And I can sort of wage these evolutionary wars within individual boxes. And so I would be able to see how geography can have an impact on this entire process. So again, like I feel like this is the time to do this is now. I feel like I'm the person to do this. I can do this. I know enough about Python and evolutionary computing that I can do all this within reason. Now, don't steal my fucking idea. Whatever. I guess I wouldn't care. Maybe I would say if you stole the idea, maybe I would spend like all the time then assembling armies of computer algorithms to fuck with you. Okay. Okay, so that's the broad view of what I want to do. So what I've already essentially built the iron framework of the toxin antidote operons. I coded that yesterday and this morning, and it works now. So essentially, let me go through and discuss what I have done. First, let me just outline like, so I have a document here. I'll pull it in view, and we could go through this. I did outline like a hypothesis. Again, don't listen to me. My hypothesis is that CI might be selected for under certain conditions or certain perspectives. So my thought is that this conclusion from Torelli in 1994, while certainly true, I've actually proven it, while true, it might be applied under certain contexts, and it might not be true under other contexts. I don't know. That's what I want to understand. So my hypothesis is that CI might actually be selected for under certain conditions. And by CI, I think synonymously as toxin antidote operons. And I want to know what are the conditions and when do they overrule that perspective of the host in the Torelli 1994 conclusion. And if I've made a mistake, write it in the comments or send me an email. We can have a private discussion. I don't have to publicly discuss it if you don't want to do that. Or if you want to publicly have a conversation, like you can write in the comments or we can do like a Zoom, whatever. Like I'm interested in learning more about this. If I made a mistake, then just call me out on it. It's not going to make me mad. So I think I can answer this with modeling. Okay, so let's start with modeling the toxin antidote operons. And the way that I'm starting with a framework from I learned in the evolutionary computing class, which is this schema theory. Okay, so this was when people were actually like developing algorithms that analyze evolutionary computing. So studying the evolution of evolving algorithms in computers. What are the patterns that they find in these evolutionary computing systems? And this was invented by John Holland, and I haven't read his books, although I did just order them on Amazon, so I need to read the books. But I have read the Introduction to Evolutionary Computing, which was a good textbook for the class. So I learned about this in the class, so I know enough about it. So essentially, like first, let me just give the basics of like schema theory. So so essentially, it's actually like it'll be really easy for you to understand actually, and which is one reason why I was drawn to this, because it's inherently obvious as to how this is how proteins work. So schema theory is essentially like if you design like strings of bits, so like 0, 1, let's zoom in on this, like 0, 1, 0, 1, 0, 1, 0, 1, and you somehow assign like a fitness for these bits. Like maybe you want them to match a certain scheme or whatever. Okay, so a schema would be like where you have a pattern. A schema is essentially like a pattern. Okay, so you have like a 0 and a 1, and they use these hashtags for like don't care values. So like if this was, for instance, like a schema that was important for in an evolutionary program for solving some particular problem or whatever, like this is a schema. Okay. That means you could have instances of that schema. So one instance of that would be this. This bit string matches that schema. Okay. This is different, but it's also an instantiation of this schema. Does that make sense? And this is actually, like, again, one reason why I really liked this, I loved this from this class, was this is how proteins work. This is actually like literally how proteins work. Proteins build domains, and in between the domains, they have a bunch of like don't care residues, which are perhaps like just like holding a spacer, or in some sense a spacer would care, but the individual value of it doesn't matter. And they string together these domains, so domain 2 or 3, whatever, to build these machines. Okay, this is actually like how proteins function. They function under the premise of this schema theory. So I can use this to essentially like model toxins and antidotes. Okay, so what I decided was I can build first a system that instantiates random TAs, toxin antidote operons, okay? And it would essentially, the way that it works is it would essentially run like this. There's some minimum value, and it might be a parameter that the evolutionary algorithm can pick itself or evolve, or it might be a parameter that I set. I'll probably allow it to evolve it in the future, but for now, you might just set it. So there might be a minimum like length of a particular like antidote. Let's say like 5 bits would be a particular antidote. And you might set a maximum value, by this I index values, so like maximum length. This should be length. So maximum length of perhaps like 5 bits. Minimum length 5 bits, maximum length of like maybe like 25 bits. That's how I have it set up right now. And then and that would be for like the toxin. And then you can do the same thing for the antidote. Minimum length 5 bits, maximum length 25 bits. So essentially I'm allowing, by setting whatever these parameters, I will allow the algorithm to pick a random size between 5 and 25 that will constitute a toxin, and a random size between 5 to 25 that will constitute the antidote. Okay? And then I can build the algorithm to randomly fill the string with 3 different values. We'll give it 0s, 1s, and 2s. So it's going to randomly construct a string of 0s, 1s, and 2s, example, something like this, between sizes 5 and 25. And one of these is going to represent a toxin. And one of these is going to represent a corresponding antidote. And they might not be the same size. because it's random, okay? And then what I can do is I can set a fitness for each pair. And here I'm making an assumption. Assume that binding efficiency, so how tightly those pairs match and how tightly they bind, assume that that binding efficiency correlates, maybe I shouldn't say perfectly, but correlates with rescue efficiency. So this is one essential kind of like assumption of the actual lock and key toxin antidote hypothesis. Okay, one actual assumption of this is that the physical act of rescue is accomplished by the binding of A to B. Okay, so if you have an antidote and you have a toxin, the rescue of CI happens by this physical interaction. And there's a lot of proof for this. There's a proof in my eLife 2019 paper where if you bind the antidote to the toxin, it actually radically structurally, well, not, I don't know if I can say structurally, but it radically adjusts the binding preferences of what the toxin is finding and interacting with. So we know for a fact that the binding of A clearly impacts what the toxin can actually like grab onto. So there's physical evidence for this, plus the fact that they bind each other. That was initially discovered in the Chen paper, Hong Li's paper in PNAS, and then my paper in 2017, the Dub paper in Nature Microbiology. So the evidence for this is extremely substantial, okay? But here's where the assumption is. This has not yet been proven that the binding efficiency correlates with rescue efficiency. So by that, what you should be able to someday test, this is a hypothesis that nobody has tested yet, is if you interfere with this binding, say you could reduce A's binding by 1/2 to the toxin, this should reduce rescue efficiency by 1/2. So you would have 2 times more embryos dying under this context if you could mutate the residues that were important for binding and sort of reduce it by 1/2. That has not been proven yet, and that is what I am assuming in this model. Okay, I'm building that into the toxin antidote framework of this model. Okay, so essentially that's why I say the fitness for each pair is going to be mathematically evaluated as essentially correlated with or equate to the efficiency of rescue. Okay, and that's a key assumption that I'm purposefully making. Okay, now how would I evaluate? So now once I have the algorithm, which I do, once I've already written the algorithm that can essentially like assemble these random toxins and antidotes, I can vary all these different parameters, and I can instantiate 10,000 of these different pairs. I could instantiate 1,000,000 different pairs, or 100 million, or 1 billion different pairs of these in an instant on computer. Okay, and then the part that takes computational time is I have to evaluate the fitness of each of these pairs. Okay, so essentially the way that I'm deciding to evaluate the fitness of these pairs is I have designed, I have built a string, essentially like a string sliding window algorithm. Okay, this is common in searching algorithms where you're searching strings. This is not anything new. I didn't invent this. Although I did sort of like I did code my own personal one, so I built it from scratch, and it's probably different from other people's. Anyway, the idea of the sliding window is if you want to match something. So say you have X, Y, Z, and you want to match it with something, A, X, Y. How do you actually do this in an algorithm? Okay, the way that you actually do this is you first it's just 2 linear scans. You align this thing up. Why did it do that? You align this thing up. You start here. Let's say this is 1 bit string. This is another bit string. You line this up, and you actually tell the algorithm, okay, line it up. Ask the question: are these equal? Does X equal Y? Okay. If yes, you tally some kind of a point or whatever, you keep track. If not, you don't tally a point, or perhaps you subtract a point, okay? And then essentially you slide. You slide this, you shift it over. So now the next analysis is A, X, Y, okay? And now you can see, now you do the analysis on after the first slide, and now you see you'd have a tally of like 2 points. X matches X, Y matches Y, okay? And then you slide it, and you progressively slide it more and more and more. And this algorithm was actually kind of difficult to write. So it was easy to write the algorithm that initially, I gotta change this. It was easy to write the algorithm that initially I could line it up like this, okay? And then I would check the values, slide, check the values, slide, check the values, slide. And then when I got to this position where the indexes matched, then my algorithm like would stop functioning, because sort of like the indexes that you're comparing starts to get wacky, because as you slide one more, here your base is always the 0. Now after you slide this one past the edge of that so that they're overhanging, now your base is no longer 0. So you have to design a system that realizes when this has happened, and then you adjust the incrementation. So maybe I don't have the most efficient algorithm. It's about like 30 codes long, or 30 lines of code, the algorithm that compares these. But essentially, like, that's literally all it does. Is it takes the longest one, the longest bit stream, whether it's the toxin or the antidote. So imagine you have something like this. It takes the longest one and puts it on the top. It takes the short one, something like this, whatever. And then all it does is it slides it all the way. And every time it slides one position, it checks every value. So what this is in terms of computer science, this is there's actually there's terms for this. This is a linear scan. It's a nested linear scan. It's a linear scan inside of a linear scan. That means like if you have a thing of numbers, a string, a bit string, it scans 1, 2, 3, each of the numbers. So essentially, like, as this thing slides, the first linear scan is the sliding to each position. The second linear scan is at every position, you need to check the comparison of every bit. So it's a linear scan within a linear scan, which means that the big O is n squared, something like n squared. So the algorithm is not, it's pretty efficient. I mean, it's not like, it's not going to cause me problems. Although it might in the end when I start doing like a whole bunch of comparisons. But for now, like, this is good enough. I feel like this is a good enough algorithm. So what it does is it compares the bits, and there's a couple questions. If 0 is equal to 0, then you get a point. Okay, so this is how it's analyzing the fitness of the toxin antidote operants. If 0 is equal to 0, you get a point. If 1 is equal to 1, you get a point. Okay, if 0 does not equal the other thing, so essentially when 0 does not equal 1, or whatever, when they are mismatch, you get -1. So this simulates affinity in the actual protein. This is how I'm simulating 2 amino acid residues binding each other through an affinity, okay? And this simulates repulsion. So you can imagine if you had 2 oppositely charged amino acid residues, they would not just necessarily like not attract, they would actually repel. So I'm simulating both affinity and repulsion in these bit strings and through this comparative algorithm, okay? And then I also have 2s. So a 2 is equal to a don't care. A 2 is essentially like if you compare a 1 to a 2, it's just a pass. Or a 0 to a 2, it's a pass. Or a 2 to 2, it's a pass. So it allows the toxins and the antidotes to build a schema. This is how the 2s are how I'm assembling these schema. Okay, so it builds a schema, or schemata in plural, and so you're going to assemble things that might look like locks and keys, right? And maybe the key looks something like this, something like this. This would be a good match. It matches right here. Okay. And the tally would be 1, 2, the max score of this thing would be +3. Okay, so then I can instantiate 1,000,000 of these toxin antidote pairs. I quickly evaluate their fitness, get the maximum fitness, okay? And then I can select them based on like fitness. So the first thing that I'm going to test is not super informative. The first experiment is essentially like instantiate a population of 10,000 TA systems, evaluate their fitness, and then like start evolving them, okay? So that means there will be, I haven't built this yet, but there will be, so the first class in Python is a TA class, okay? The second class is going to be the evolutionary algorithm class, which runs the evolutionary program. So in that class, there are various methods, which are going to be parent selection, so fitness proportional selection, truncation, K-terminant. I'll talk about all these things later. There's parameters within the evolutionary algorithm, so they might be able to evolve the max length, the minimum length, within some kind of a constraint. You can't have infinite length, because obviously if you could have infinite length, you would just evolve an infinitely long antidote, and it would obviously match somewhere. Now, in biology, you can't do this, because you have a constraint of it costs energy to replicate this. So there will be a penalty. So somewhere in here, I'm going to build a penalty for the length to prevent infinite long, so there's going to be a fitness penalty. This is important. There will be a penalty function that gives a fitness penalty for length. So once you evaluate the fitness, the best matching pairs, there will be some kind of subtractive coefficient value that will give a penalty for how long those things are to simulate the biology that these things are not infinite resources. They cannot get infinitely long. Okay, and then the EA algorithm class, which we're talking about. So the way that this works is you instantiate a population. So you actually have like it's like population biology, but in a computer. It's fascinating. Instantiate 10,000 different TA systems. We'll start with 10,000. Essentially evaluate their fitness, select parents. So you take parents that have good fitness, okay? So after you've analyzed all the fitness of all the TA values, you pick pairs and you actually mate them together. Okay, and mating, the reason that biological organisms mate is twofold: is recombination. That's literally like the purpose of sex, and mutation. Okay, so the way that you build genetic diversity and get genetic diversity over generations is through mating events. Okay, and so every time a mating happens, there's going to be some probability for a recombination and some probability for a mutation. And you might think as a biologist, well, what's recombining? What's mutating? And the answer is actually the chromosome. The chromosome is, there's actually going to be two chromosomes, chromosome 1 and chromosome 2. And chromosome 1 is simply going to be the T, the toxin. Chromosome 2 will be the antidote, and the chromosome will also contain a few different things. So it will contain the bit string of the toxin, the bit string of the antidote. It will contain the parameters that control these things. So that there will be the length, the length of the toxin, the length of the antidote. So that will be able to mutate, to change. Okay. What are my other parameters? Oh, the mutation rate, the recombination rate. So these things will actually be able to essentially like pick how much they want to recombine, pick how much they want to mutate within a generation. And then after the mating happens, you do the mutations, you do the recombinations, and you instantiate then new sons and daughters, like sex is gender doesn't matter here. You initiate new offspring, okay, that then are new instantiations of either the old schema or new instantiations of a slightly modified schema. And then the children have a chance to have a better fitness than their parents. And so then you evaluate the fitness of the children, and then you do what's called children selection or offspring selection with various methods, which I'll talk about later. So then you select the offspring, okay? And then after you select the offspring, you essentially figure out a survival. Like who survived to the next generation. So let's say the population has a carrying capacity of 10,000 individuals. That's the max population. And if I give every a certain amount of parents a certain amount of offspring, that means it's going to be 10,000 plus offspring. So you have to build in a death rate. So you decide who dies, and typically there are different ways to decide who dies. But essentially, you're mimicking like an ecological system that has selection, that is killing off these things that don't fit together properly. So the goal here is initially start off easiest experiment, evolve a bunch of TAs, kill the ones that don't match very well, and see what particular schemas evolve. Okay, and what I expect to see, here's what I expect to see, is I expect to see one of these things will get very large and one of these things will get very small. So it's likely that the toxin will either get large or small, and it's likely that the antidote will either get large or small. And it's likely that then the reason for that is because you're more likely to match in a good sense if you have sort of a longer thing to like grab onto. Now here's where it's going to get interesting. That's experiment 1. Okay. Experiment 2 is where it's going to get really, really interesting. And it's going to start to be informative. So from a toxin antidote's perspective, from the perspective of the TA, okay, you want a perfect toxin that kills at 100% efficiency. And you also want a perfect antidote that rescues at 100% efficiency. This is going to be the strongest selfish genetic element, the strongest driving construct. And so the values are going to converge on that in experiment 1. Here's where it gets interesting in experiment 2. In experiment 2, I'm going to make them compete with each other. I will make the antidotes and toxins compete for fitness amongst themselves. So that means when we talked about the sliding algorithm that evaluates their fitness, now what I'm going to do is I'm not just going to take one TA pair and slide it with its cognate pair and calculate the fitness value. What I'm going to do is I'm going to take that particular instantiation of that antidote and I'm going to compare it against all the other 10,000 toxins. And now what a TA system wants, the best TA system will rescue all other toxins, but have a toxin that no one else can rescue. So what you want is a super unique toxin that nobody can figure out the schema towards, that nobody can unlock it. And you want a toxin that can unlock everything, because that's going to be the best combination. That's how these selfish genetic elements are going to compete. So experiment 2 is going to be start competing selfish TAs against themselves. I'm super excited for that, and I'm really close to being ready to do this experiment. So now you guys understand the premise. This is where I'm starting. Recognize that what I'm doing is I'm building models to represent the biology of how these different levels function. So I'm starting with this. Once I have this built and I understand the principles of how the TA systems work, then I can start building in these subsequent levels all the way up to the level of like ecological geography. And it's going to be fascinating to see how this all fits together. So I've got the first level built. I'll probably run the experiments next week and look for an update video sort of like reporting the results of what I found in these experiments. Again, I don't think you're going to see a lot of interesting results in experiment 1, but it's going to be when we start evolving these things in experiment 2 against each other, competing with each other, that you're going to start to see, I think, some interesting things. And the other final thing I would say is if there's anything I learned from the evolutionary computing class, it's that humans cannot predict evolution. So the idea that like, oh, we're going to make this hypothesis and we're going to like try to figure out like how to understand just through thinking like how these 6 different levels are going to function together, that's never going to happen. So using the computer to simulate it and clearly like reporting the results is going to help me better get a grasp of how these things are functioning. Okay, so with that, I will end. This is going to be a fun research project, public research project. Don't steal the idea, but you can participate if you want. Write comments, criticize, tell me why I'm wrong, tell me why I'm right, tell me what you like, tell me what you don't like, send me emails, do whatever you want. Who cares? Who gives a shit? Okay, I had fun. See you around next time for the update.
I shall meet death head on. Entertain